I'm going to start by giving a short biography. I did my medicine in India in a place called Gittleman, then in 1992. Then did my MD and then moved to the UK, did my PhD, and then worked as a consultant. And my research has been in the genetics of hypertension ever since 1999. And I did a short stint at the Broad Institute in 2010. So the outline of my talk will be around a short introduction to precision medicine, hypertension genomics, and I'll describe a gene and a metabolomic marker for hypertension. And I'll end with some insights from a blood pressure clinic, which is one of the largest in the world, with a lot of outcome data and some future directions. So when we talk about disease, we talk about rare diseases and we talk about complex traits. And as this is a sequencing company, most of your experience would be with monogenic syndromes. I deal with complex traits which come in the middle. They are multifactorial. They are due to multiple genes and multiple environmental factors working together to lead to a phenotype. It's not a single gene condition or a single environmental factor. So that makes it complex. I mean, we call it complex diseases, and the analysis of these are pretty complex. And I work with hypertension, and the reason why hypertension is important is reflected in this picture. This is the WHO Global Burden of Disease Survey. This tells you, this gives you the burden of mortality. What are the major risk factors that contribute to the most deaths worldwide? And the highest is high blood pressure. And this contributes to the maximum number of deaths, the single risk factor, which contributes to the most death worldwide. It's not a problem with developed countries. It's a problem both in the developing and the developed countries. When you convert this to numbers, 1 billion people worldwide have high blood pressure, and majority of them, their high blood pressure is undiagnosed. Contrast this to diabetes. There are about 450, half the number of people with diabetes. So huge numbers, and because high blood pressure leads to an increased risk of heart attacks, strokes, kidney failure, the morbidity associated with high blood pressure is huge. And then these, if untreated, these lead to early deaths. And the biggest problem, it's called a silent killer because there's no symptoms until you present with a stroke, and then they recognize that you have been having high blood pressure for a while. So a lot of problems in terms of early diagnosis, identifying the right patient, and as we'll come to later on, problems with treatment. So we are all clinicians, we practice medicine, and the type of medicine we practice is the traditional way wherein we make a diagnosis, we see a patient, do a few tests, make a diagnosis, and once we define the diagnosis, we give the same treatment to everybody. And some patients will respond to the treatment we give, some will not respond, some will develop side effects, but it's all a trial and error process. And this is not new. We've known this for a long time. This is William Osler in the early 1900s who said, because of this big individual variability in treatment response, we are actually practicing an art. The medicine we practice, though we claim that it's a science, it's mostly an art because there's a lot of experience and other factors which go into the current practice of medicine. And what about the treatments that we have? These, as I mentioned, these are common diseases. Common diseases or complex diseases which are extremely common, they affect a large proportion of the population. So these are public health problems. And we have drugs to treat many of these conditions, but most of these drugs are ineffective. If you look at commonly prescribed drugs, 38% of antidepressants do not work 38% of people who are prescribed antidepressants will not benefit from the drug. And it varies. If you take diabetes drugs, 43% of individuals will not respond. And for cancer drugs, up to 75%. So there's huge amount of drug non-responsiveness in current clinical practice. Again, if you want to look at numbers, these are some of the commonly prescribed drugs in the US. This is your Nexium, your commonly prescribed antacid. you need to prescribe it. One person, if you prescribe it to 24 people, one person will benefit. Similarly, rosuvastatin is a drug for high cholesterol. You need, for every person who responds, there's probably about 20 people in whom the drug would be non-responsive. If you take some of the newer biologics, then the numbers needed are smaller. So that's etanercept, the anti-TNF blockers, or infliximab for Crohn's disease. You need to treat about 4 for one person to respond. So there's a big problem. And that's where this whole concept of precision medicine has come, mainly to try and target the right drug to the right patient. But more specifically, precision medicine is to use the information about the individual's genes or metabolite profile, proteomic profile, any of the omics profile, use those to prevent, diagnose, or treat disease. So we need to combine all these together and try and improve health. And the driver for this has been the enormous success with cancer. Because for cancer, you are already practicing precision medicine. Cancer, of course, is an important disease. It affects a proportion of the population. What we want to do is to try and translate the successes of precision medicine and cancer to common diseases. The reason for that, as I mentioned, a big problem with unresponsiveness, which imposes a big burden on the healthcare system, not only on the economic side, but also on the patients, because patients get burdened with drugs which don't work, burdened with the side effects, and they do not benefit from the advantages the drugs would offer if they worked. So where does this all come in? For common diseases. So what we'd like to do is, for precision medicine to work, you need to redefine the current taxonomy of disease. Now, when you come to my clinic, I'll put a cuff on your arm and diagnose you as having high blood pressure, and then I just give a series of drugs. That's just an umbrella term for all the different pathophysiological pathways that lead to high blood pressure. You could have high blood pressure because your sympathetic system is not working, or you're retaining salt, or you got oxidative stress. Various mechanisms work together with your dietary and other environmental factors. The classification, and that's the same thing for diabetes, for any other disease. The classification system goes back to 1855. This is from England. William Farr classified disease, and we are using the same classification system. It's not changed. It's based on symptoms, it's based on an organ, and it's based on some phenotypic features. There's no molecular or functional characterization used in the definition or labeling somebody with a disease. So obviously, and this is very obvious, you know that if you label somebody with an umbrella term, you may just prescribe them treatment blindly, and so patients may not benefit, or if you diagnose wrongly, then you're going to give treatment to a patient or expose patients to drugs, which has got its associated risks. And these are some examples. If you think of diabetes, type 1 diabetes is actually multiple diseases. It could be due to problems with HLA, it could be autoimmunity. Type 2 diabetes, we already know that it's a heterogeneous phenotype. You've got, and we know certain mutations, different mutations which lead to type 2 diabetes. And breast cancer, as you know, in terms of cancer, we are actually redefining cancer based on mutations, because all these mutations are druggable, and hence defining a cancer by the molecular signature means that you can treat them properly. And similarly, on the other hand, you can have one mechanism which can be implicated in more than one disease, and this is just one example. Ciliary defects can lead to various problems. If you know the mechanism, then you can decide on a tailored treatment. But the challenge is to get this sorted, to get the mechanism sorted. And this must be well known to you. This is how the lung cancer. So the traditional view originally was large cell, squamous cell, and adenocarcinoma, and now we have all these mutations. So we have actually stratified a disease. So William Farr's definition of lung cancer would be that one, and that improved with histology, and now we've got genomic diagnostics. And the advantage of genomics come from, so the advantage of genomics in precision medicine comes from cancer predominantly, and most of the cancers that you know of are due to genetic mutations. A major proportion of the genetic mutations are druggable. So the question is, how do we tailor treatment? Yes, we can identify the genetic mutation, but what has been the effort so far? We have, for common diseases, we've tried looking at the pharmacokinetic aspects, I mean, drug metabolism. Perhaps you do not respond to a drug because you do not absorb the drug, you don't metabolize it. Many of the drugs could be prodrugs, they need to be activated. So if you lack the specific cytochrome P450 enzyme, you won't activate the drug, so you may be non-responders. All this makes good sense. You've got chips which could genotype all your cytochrome P450 polymorphisms. Doesn't work because we've tried a lot. It doesn't work. Pharma companies are very good at doing the initial screening of big pharmacokinetically affected drugs and take it off the pipeline. So this has not had much impact. There's probably better impact with differentiating disease. So rather than differentiate people, what you'd like to do is to differentiate disease, and that comes back to redefining the taxonomy. So that's the overview of precision medicine, and we'll just now focus on hypertension. 1 billion people, that means the prevalence of hypertension is 27%. So 1 in 6 of you will have hypertension. High blood pressure is, that's overall among adults. 27% of adults have high blood pressure, but that's age related. Your blood pressure increases with age. So at the age of 60, about 55% of adults over the age of 60 will have hypertension. If you go to the age of 70, then almost 80% of you will have hypertension. So if you live long enough, everybody gets high blood pressure. And what is it about high blood? What is the level at which high blood pressure causes risk? Now this is a study wherein they've accumulated data from all the clinical trials, almost 1 million people, and they just looked at outcomes. And what you see here is this is the risk of death. And they've done the analysis for various decades of increasing decades of life. And what you see is a linear relationship. It's on the log scale. So you see a linear relationship between blood pressure and mortality. So this is mortality due to stroke, and on the other side is mortality due to heart attacks. And what you find is there's no cutoff. You have a linear relationship. When you go to your doctor, you will be told that you have a blood pressure of greater than 140/90, you've got high blood pressure, we need to start you on treatment. And that's the threshold we use to start treatment and reduce your blood pressure. But if you look at the data, the risk is continuous. For every point, every millimeter change in your blood pressure, you have increased risk. Of course, if you're older, you have higher risk, you can see, but the lines are parallel. The risk is continuous from about 115. That's extremely low blood pressure. From about 115 millimeters of mercury systolic blood pressure, you have increasing risk. Does reducing blood pressure improve survival? Yes. Again, from the same study, even at 2 millimeter, it does not matter how much your blood pressure declines. Even if you reduce your blood pressure by 2 millimeters, that translates to big improvements in heart attack and stroke deaths. So you reduce your blood pressure by 2 millimeters, you have a 7% reduction in heart attack deaths, a 10% reduction in stroke deaths. Now, high blood pressure does not occur on its own. It comes with a lot of baggage. So if you have high blood pressure, it does not mean that you only have high blood pressure. It marks you as somebody with high cardiometabolic risk. It marks you as somebody who has slightly high cholesterol. It marks you as somebody who will be slightly obese or overweight. It marks you as somebody at a higher risk of getting diabetes. So when you treat high blood pressure, you don't treat the number, you treat everything else. You need to check for everything else. We check what's called the global cardiovascular risk. And we know that if you treat everything, if you treat multiple risk factors, there is more than an additive effect. So if there's a slight reduction in your blood pressure, plus you add a 10% reduction in your cholesterol, that translates to a 45% reduction in cardiovascular death. So even small changes, making small changes that work in multiple risk factors will translate to big improvements in survival. So So that's what we know, and that's the evidence we're using. High blood pressure has been known, but what causes high blood pressure? This has been described even as early as 2600 BC. And the Yellow Emperor of China, that's a book that described high blood pressure, that described the relationship between salt intake and blood pressure, and all the complications, heart failure and edema and all that. The word hypertension was defined sometime in the early 1900s, and this was the relationship between high blood pressure, or the high blood pressure was defined as a disease primarily after evidence not from doctors but from insurance companies. And this is what happened in New York and in the US. The insurers like to measure various parameters, and you had a cuff to measure blood pressure, so they started measuring blood pressure, and they just wanted to calculate your premiums. And they found that blood pressure related to early deaths. So they were actively using it before the doctors, physicians came in, and then you had, it required evidence from the Framingham Heart Study and other related cohorts to link high blood pressure as a significant predictor of heart attack, stroke, and early deaths. But it was the insurers, the actuarial tables actually correlated high blood pressure with risk. Again, nobody knew what caused high blood pressure. At some point, people thought that high blood pressure was essential. We call it essential hypertension. And the reason we call it essential hypertension was because people recognized that those who had high blood pressure and those who developed stroke, their arteries were quite thick. They could feel the hardened arteries. And when they dissected, they found that the arteries were narrowed. So they thought that that high blood pressure was actually protective. Because you had these narrowed arteries, you need a higher pressure head to push blood through your blood vessels. So they considered high blood pressure to be essential, and it was not pathologic. Your narrowed arteries was pathologic, and you needed the blood higher pressure to go through it. But now we know that it's the other way around. It's the blood pressure which makes these arteries narrowed. This was 1960s. Page actually recognized that this your blood pressure is regulated by multiple factors. It was genetics, hemodynamics, humoral, endocrine, neurological, adaptive, anatomical. All these interacted together to lead to a phenotype, which was what we measure using a blood pressure cuff. In the 70s, this is Guyton's model, again from the US. This is a circuit diagram. He tried to develop a fancy diagram about how blood pressure is regulated. And this required multiple segments. You required your brain, your circulatory system, your kidneys, your heart. And even now, that's what we know. This is done in 1972 or so, and we still have recognize all these physiological pathways that regulate your blood pressure. Again, in the early 70s, there was a big debate. Is high blood pressure a separate disease or something else? A big debate between Platt and Pickering in England. Basically, it depends on how you look at the condition. Platt basically measured blood pressure in people who had high blood pressure and people who had low blood pressure. And then he said, blood pressure is a separate disease. You either have high blood pressure or you don't have high blood pressure. Pickering instead measured blood pressure, and he looked at the distribution, and he found it was normally distributed. So he said it's not a separate disease. It's just that you are at the upper end of the distribution, and that's and whatever threshold you use as a cutoff, you will label somebody as hypertension. Now, if Platt was right, if there were two populations within the population, one with high blood pressure and one with normal blood pressure, then you'll see a bimodal distribution of blood pressure in the population. We don't see that. We see a normal distribution. So, and the basis of a normal distribution, if any phenotype, if any trait is normally distributed, that means you should have hundreds of factors contributing to the variation in the trait. Only then you get this smooth curve. So that means it's a polygenic multifactorial disease. Genetic component, what's the evidence for genetics? This is a nice study looking at the correlation of blood pressure between spouses and children, between natural siblings, adopted siblings. And they showed higher correlation if you share some genetic material. So you've got a higher correlation of blood pressure between natural siblings and between parent and child, but very little correlation between parents and adopted children. So This would indicate that something which is transmitted within the family, which is making, which results in this higher correlation with blood pressure. And what is transmitted could be, it could be the environment. You're living in a house, and you're sharing all the behavioral and diet and everything. So it could be environmental. But when you look at adopted siblings, you don't see that the excess of environmental factors contributing to the correlation. So this has to be genetic. So it's about 38%. When you calculate heritability of blood pressure, it varies between 45 to 60%. So you've got heritability of blood pressure. There's evidence from these studies which show that genetics could contribute to this. And you've got data from monogenic forms, the rare syndromes of high and low blood pressure, which are due to single gene mutations. This actually gives you the evidence that genes can perturb your blood pressure. A single gene mutation can affect your blood pressure. Of course, what's interesting is most of the genes that we've identified for blood pressure syndromes are all based on the kidneys and a few in the adrenal gland. And some of these mechanisms are quite interesting. If you look at glucocorticoid remediable aldosteronism, now this is a rare syndrome. Most of the monogenic syndromes present with high or low blood pressure in childhood. Now, glucocorticoid remediable aldosteronism is a problem due to a chimeric gene. So you have two genes sitting very close together in chromosome 8. That's the aldosterone synthase gene and 11-beta hydroxylase. They've got almost 90% homology. Probably these two genes have arisen from a gene duplication event in the past. But aldosynthase produces aldosterone, which is the key enzyme to retain salt. So that's secreted by the adrenal gland in response to angiotensin II or a low potassium or low, and what stimulates that is low blood pressure. So if you have low blood pressure, it triggers the renin-angiotensin system from the kidneys, and angiotensin II will stimulate aldosterone. It makes you retain salt and get your blood pressure up. 11-beta hydroxylase is the gene which produces cortisol, your steroid hormones. So it's a stress hormone. And the regulation of 11-beta hydroxylase is from a pituitary. You secrete adrenocorticotrophic hormone, which gives you, which regulates your cortisol. There's just a diurnal variation in your cortisol secretion. So though they are homologous, they have subserve two completely different pathways. Now, because they are very close together and they got a high sequence homology, during meiosis sometimes you can get unequal crossing over, and you get a chimeric gene. So what happens with the chimeric gene? It's got a bit of your aldosynthase gene and a bit of your 11-beta hydroxylase gene. The 11-beta hydroxylase bit is the one which responds to adrenocorticotrophic hormone, and you've got the aldosterone coding part there. So you've got the aldosterone gene segment, and you've got the ACTH responsive segment. So now you've got a gene which produces aldosterone in response to your pituitary signal, not from your blood pressure. So you get constant stimulation of aldosterone irrespective of your blood pressure. You retain all the salt and your blood pressure goes up. So the treatment for this, if you know this person has GRA, the treatment is to give them steroids, because the best way to block your ACTH is to give steroids, biofeedback, and it blocks your ACTH and your blood pressure comes down. Liddle syndrome is another interesting mutation. This is due to a mutation in the epithelial sodium channel, which is the main sodium reabsorbing transporter in the collecting duct of your nephron. And this is the main pathway by which aldosterone reabsorbs sodium. Now, if you have mutations in the epithelial sodium channel, so what happens in the epithelial sodium channel? It sits on the cell surface. It's in dynamic equilibrium. Your channel gets internalized, and then you have a constant number of channels on the cell surface. Mutations in the alpha or beta subunit results in, so the internalization is through a WW domain here on the cytosolic side of the transporter, and that leads to the transporter getting internalized. In Liddle syndrome, you have a mutation that results in this recognition site being lost, which means your epithelial sodium channel persists on the cell surface. You've got lots of epithelial sodium channels, all of them reabsorb sodium, and you get high blood pressure. If you know that, treatment would be a specific blocker of ENaC, and that was amiloride. It blocks ENaC and it reduces your blood pressure. Apparent mineralocorticoid excess is another syndrome. So as I told you before, aldosterone is a mineralocorticoid. It basically reabsorbs sodium. Cortisol, as we know, does not affect sodium balance. That's what we know. But intracellularly, cortisol actually is a very good mineralocorticoid. Cortisol is as effective as aldosterone in reabsorbing sodium. But what happens in the cell, you've got an enzyme called 11-beta hydroxysteroid dehydrogenase, which converts cortisol to cortisone, which is not a mineralocorticoid. Now, if you have mutations in this gene, this means cortisol persists intracellularly, and it will act on your epithelial sodium channel and reabsorb sodium. And if you like licorice, if you eat lots of licorice, licorice contains glycerinic acid, which is a blocker of 11-beta hydroxysteroid dehydrogenase, and you get hypertension. And you stop that, then you can revert your hypertension back. So you can either have mutations in this, or if you eat a lot of licorice, you block that, and cortisol behaves like a mineralocorticoid and gives you hypertension. Then we are moving to the other bit of the nephron, which is the distal convoluted tubule. The distal convoluted tubule, you got a syndrome called Gordon syndrome, which is due to mutations in the NCC channel. The NCC channel is a major sodium transporter in the distal convoluted tubule. You can have mutations there. You could have mutations in many genes which regulate NCC. So that's Wink4, Cullen3, Kelch3. Mutations in all of them cause a syndrome which looks like which is a salt retaining syndrome, and you treat it with blocking NCC, which is thiazide diuretics. So thiazide diuretics like benrofluozide, chlorthalidone are specific blockers of the NCC channel. Gittleman syndrome is again a syndrome associated with transporters in the DCT. This is a milder syndrome, and this is not a high blood pressure syndrome. This is a low blood pressure syndrome. You may know people who will have very low blood pressures. They'll say their blood pressure is 90 systolic, and they'll be healthy. These people may be heterozygous for Gittleman syndrome. It's quite frequent. It leads to salt wasting, a little bit of salt wasting. So these are people who like salt, who crave salt. They will have salty crisps. They'll eat a lot of salt, but their blood pressure will never increase. They maintain their blood pressure by eating a lot of salt. If they don't eat salt, they become dizzy and they have low blood pressures. So that's Gittleman syndrome. It's common. It's about and what you see would be the heterozygous state. So people who have low blood pressures and they eat a lot of salt, that could be just due to a mutation here. Again, this leads to this classic story of confounding. If I were to prove that salt increases your blood pressure, and I don't consider this variant, then I would study a population and say that, oh, all these people are eating salt and they don't have high blood pressure, so salt does not increase high blood pressure. But if you had known the mutation, then once you correct for that, then you'll know actually salt increases blood pressure. So understanding the underlying genetic variant can actually make sure that we make the right interpretation of our data. This is another low blood pressure syndrome called Bartter syndrome, and this is in the mutation in the thick ascending limb of the loop of Henle. Now this is a segment of the nephron which is only permeable to sodium. It is impermeable to water. And the transporter there is NKCC2, and this segment reabsorbs 25% of your filtered sodium. So it's an important part of your sodium homeostatic pathway. Mutations in many of these genes, NKCC2, chloride channel, ROMK, which is the potassium transporter, there's a calcium sensing receptor here. Any of these can lead to salt wasting and low blood pressure. So that's a monogenic syndrome. So we are now pretty convinced that genetics does have a role in regulating your blood pressure. But the problem with all the syndromes that I've told you is they are at this end of the spectrum. They're monogenic, they're due to single gene variants. They are very rare in the population. They are of no public health importance. they're very good in allowing us to understand biology of blood pressure regulation. We are interested in essential hypertension. That's the 1 billion people with high blood pressure, contributing to about 52 million deaths worldwide per year. And it's a complex trait. As you know, you've got a lot of factors which contribute to blood pressure. Those are the environmental factors. You've got multiple physiological pathways, and some combination of those factors will lead to high blood pressure. So it's a big problem. The genetic part of that, yeah, there has to be some genetic component, but these are polygenic, so you're looking at multiple variants with very small effects all adding together to lead to the phenotype. So we want to find, because it's a common trait, we would expect common variants to be associated with the disease. So that's the whole common disease, common variant hypothesis. And the first genome-wide association study was the WT's, the Wellcome Trust Case Control Consortium. And you may be familiar with this. This is the standard Manhattan plot. You've got the genomic coordinates on the x-axis and the negative log of p-value on the y-axis. So that means the higher you go up on the y-axis, the lower the p-value. And what we're looking for are signals which cross a genome-wide p-value threshold of 10 to the power of -8. And so this was a great study because they looked at 7 common diseases, and they showed for the first time quite strong signals in a hypothesis generating manner for various diseases: coronary disease, Crohn's disease, rheumatoid arthritis, and diabetes, but nothing for hypertension. And so this was a big problem. There was no signal for hypertension. The problem was that the Wellcome Trust at this point decided that hypertension was not a genetic disease, and they said they're not going to fund any more research into hypertension genetics. But the problem why we had no signals was mostly a design problem. Because this was such an expensive and complex project, what the consortium did, they selected all the case cohorts, and they had a group of non-phenotype controls. So each case cohort provided 2,000 cases. And you had a pool of 3,000 individuals who formed the controls. These were blood donors. There was no phenotyping. And based on power calculations, it was clear that these were well powered to detect signals for all these diseases. But the problem with hypertension is, as I mentioned, 27%, a third of you will have high blood pressure. So 30% of the controls were misclassified as controls. They could have been hypertensive, and that diluted power, and we didn't see that signal. Following that, large consortia formed, and we've got a lot of signals for signals for blood pressure regulation, some signals for hypertension. And this is the current genomic landscape for blood pressure. There are now over 100 SNPs associated with blood pressure. So we have now moved on from the Page model of 1960. Now we've got a deeper understanding, slightly more understanding of various pathways that affect blood pressure. We've got a lot of monogenic genes, and we know which pathways those genes affect. So we can update the Page model with this. So it's still not a complete understanding of the phenotype, as I'll come to later. So the other aspect is treatment. So we are understanding a lot of blood pressure regulatory mechanisms. We are discovering genetic variants. What about treatment? Again, if you go back to the timeline, treatment for high blood pressure initially consist of various things, but most of the drugs that we commonly use were discovered between 1950 and early 2000. There are no new drugs in the pipeline for high blood pressure. And most of these drugs were developed through an understanding, or by accident, or through an understanding of physiology. So we know that salt increases blood pressure, so if you have a compound that makes you lose salt as a diuretic, then that reduces blood pressure. So all these drugs were discovered through an understanding of physiology and not of genetics. And this is just reflected here. So these are all the monogenic syndromes for high blood pressure. And this is when the syndrome was described, and the arrowhead indicates when the gene was mapped. And what you find is none of these genes, despite these genes giving us quite a detailed understanding of blood pressure regulation, none of these genes informed drug development. So that's where we are here. We are here at the post-GWAS era, where we've got hundreds of SNPs, thousands of SNPs being discovered for various traits. And this is the time when we would expect these genomic variants to inform new drug development. So I'll focus on the GWAS, which I've been part of, and take you through the process. So this was published in 2010. So given the problem with the Wellcome Trust Case Control Consortium, we decided to follow that route, but design properly. So there was a split. Most of the blood pressure researchers decided that the best option would be to create a large cohort. Secondly, instead of studying hypertension, study blood pressure as a quantitative trait, because you're statistically more powered. It's a better phenotype. So they decided. So they joined together and created a consortium with up to 100,000 people, got the phenotype, and then they did that, and they were quite successful. We were deeply looking at the Wellcome Trust data, and we thought there was a big problem. The problem was this misclassification. So we decided that if we resolve the misclassification problem, we may find something. So we decided to do an extreme case control study. We we got a clinical trial data from Sweden, and we selected people whose blood pressure was greater than 160 by 100, and that corresponded to the top 2% of the blood pressure distribution. So quite extreme hypertensive. But our problem was not hypertension. As you know, our environment is highly salt rich. We eat a lot of salt. We are very stressed. So the default phenotype right now is high blood pressure. Everybody is being pushed to high blood pressure. Under this environment, if you have low blood pressure, then that's actually an extreme phenotype. So we decided the phenotype we'd like to study would be low blood pressure rather than high blood pressure. So we had to screen over 40,000 people. So we collaborated with a Swedish cohort, the Malmo Diet and Cancer Study. They had screened 40,000 people. They had blood samples stored. We looked at their blood pressure, and we selected people over the age of 55 with blood pressure less than 120 by 80. And again, to make it very sure that these were not, again, to remove any misclassification, we also had 10-year follow-up data, and we made sure none of these individuals had heart attack or stroke over the 10-year period. So these are, we call them hyper controls. Now, to give you an example, if you're 55 years old, your blood pressure will be about 140 by 85 or 145 by 80. That's your normal blood pressure, because that's an age-related rise in blood pressure. These people had blood pressures of 120 by 80. So they're extremely low blood pressure people. So we did that. So we thought our design was good. We did all that. We thought we may have enriched for some monogenic forms. But we got a signal in chromosome 16, which didn't reach genome-wide significance. But then we went back to all the other cohorts and gave them our study design and told them, do the analysis for the top SNPs. And they did for our top SNP, and the p-value reached 10 power minus 13, which was genome-wide significant. And this is the forest plot for all the various cohorts which did our study. And it was quite good. All the cohorts, except one cohort, it was all in the same direction. This was our discovery cohort. The effect size was much larger than the final effect size, and that's basically what we call a winner's curse. Whoever does the first study will actually see an inflated effect size. So you'll think as though your SNP has a very large effect, and that's very commonly recognized. A winner's curse, your true effect size, regression to the mean, will actually be much smaller. So that's what you see here, and that's the regional plot, which I will show here. Yep. So this is a GWAS SNP, which was in chromosome 16, and that was somewhere in the upstream end of the Euromodelin gene. There are a few genes on either side. We now need to link this SNP to the gene, and when we looked at UMOD, we realized that UMOD is a protein which is only expressed in the thick ascending limb of the loop of Henle and nowhere in the body. So that was good, because if you remember, all the monogenic forms of hypertension had genes in the kidneys or the adrenal gland. And now we did Divas, and we found a SNP which was near a gene in the thick ascending limb, so that made sense. And so we were lucky because it was a gene which was expressed in the kidney, and secondly, it was a gene which shed the protein in the urine. So we could measure uromodulin in the urine, so we could relate the SNP genotype to UMOD by just measuring UMOD in the urine, and we related that in 2 studies. We measured urinary UMOD, and this was our top SNP, and we showed a significant relationship between genotype and UMOD excretion. The low UMOD genotype is the allele, so the minor allele. So the GG genotype, which is the low minor allele, is associated with low blood pressure. So low UMOD, low blood pressure, and the UMOD increasing allele is associated with high blood pressure. So that's the major allele. So the minor allele frequency is about 18%. So that means 67% of you will have at least one A allele. This is a study we collaborated with a group in the Netherlands, and they did a salt intervention study. They took normal human volunteers and gave them a period of low salt and a period of high salt. Low salt meaning extremely low salt, and high salt. So we got the genotype, and we saw something interesting. These are same individuals on a low salt arm and the high salt. They crossed over from low salt to high salt. We noticed that when they were on the low salt period, their urinary UMOD excretion was related to genotype. But once they were put on high salt, it completely nullified. So there was a gene environment interaction. Eating salt actually increased UMOD excretion. So it fitted again with the salt pathway for blood pressure, the SNP, the gene, and the salt pathway. Uromodulin is the most abundant protein in the urine. So when you give your urine for testing for lab, they will centrifuge the urine. There's sediment at the bottom. They'll chuck that sediment out and measure electrolytes and protein in the supernatant. That's a urine test. The sediment is actually UMOD. It's the most abundant protein in the urine, and nobody knows what its function is. The only function people thought UMOD did was it perhaps protected you from E. coli urinary tract infection, because the UMOD protein, which is a polymer, can trap the fimbriae of E. coli. So the assumption was UMOD protected you from urinary tract infection, but otherwise nobody knew what UMOD did. There were a few monogenic syndromes called uromodulin-associated kidney disease. These were due to mutations in the UMOD, which led to a trafficking problem, ER stress, and kidney damage. And and it also led to uric acid stones in the kidney. So uromodulin-associated kidney disease was known, and most of the people studying it were urologists, because they were interested in the stone formation and things, and these cause cysts in the kidneys. So urologists tended to study UMOD, but nobody realized that UMOD was associated with any other phenotype. So now we wanted to prove causality. Okay, we showed association. We think we measured UMOD in the urine and we linked it to genotype, but we still need to prove causation. So that means you need to have a knockout mouse model, which was available. We got this knockout mice from Oklahoma. These are urologists studying UMOD-associated kidney disease. So we got the mice, we implanted telemetry probes in them and just measured blood pressure. We didn't do anything. And we found the UMOD knockout mice had low blood pressure compared to the wild type mice. And then we showed this data back to the people in Oklahoma, and they said, oh, we've been studying this for 20 years and we've never measured blood pressure. So this proved that UMOD, and this fitted in with the human studies. UMOD, low UMOD genotype had low blood pressure. You knock out UMOD in mice, blood pressure decrease. Now next we wanted to test the salt hypothesis. So we fed these mice salt. So this is wild type mice. The black bar is the blood pressure after salt, and the white bar is the blood pressure without salt. And you find that as these mice consumed salt, blood pressure went up. There's a slight bit of variability there. That's because initially these mice, they don't like salt, But once they start consuming salt, they like salt and they eat more and more salt and their blood pressure goes up. Whereas the knockout mice, they consume a lot of salt, but there's no effect on blood pressure. And this is the pressure natriuresis curve. So the best way to assess whether you're really consuming salt is to measure salt in the urine. If you measure your 24-hour excretion of urine sodium, that's a marker of how much salt you've consumed. Because it's very difficult to know whether the mice have actually eaten what we've given them. So if you plot blood pressure against your 24-hour urine sodium excretion, you find in the wild type mice with increasing sodium excretion, or which means sodium intake, your blood pressure increases. You look at the knockout mice, they consume enormous amounts of salt, but there's no effect on blood pressure. So they're salt resistant. So that fitted with our hypothesis, and that almost gave us enough evidence that the SNP that we've discovered linked to UMOD was a blood pressure gene. Following our publication, a group in Italy completely independently did a lot of work. These guys were UMOD experts. They worked on UMOD associated kidney disease. So they did a lot of work to look at blood pressure and kidney disease. And so whilst we worked on a knockout model, they had transgenic knock-ins. So they overexpressed UMOD in mice and they showed blood pressure increased. And they also looked at NKCC2. So the thick ascending limb, the transporter is NKCC2. So they looked at NKCC2 and they showed that if you overexpress UMOD, your NKCC2, phosphorylated NKCC2 increased, and some of the regulators of NKCC2 also increased. So they linked UMOD to NKCC2 function. And the reason they got published in Nature Medicine was because they did all this biology, and they had a cohort of untreated hypertensive patients who were treated with, so this was part of a salt balance study wherein they had taken these individuals, newly diagnosed hypertensive individuals, not on any treatment, infused normal saline, gave them a liter of normal saline, and then gave them furosemide, which is a blocker of NKCC2, just to see how they were testing how well your kidneys can excrete excess salt. They had DNA on them, so they genotyped them, and they found that the individuals with the UMOD increasing genotype were better responders, showed a better response to furosemide. And that linked UMOD genotype, UMOD production to NKCC2, because furosemide is a specific blocker of NKCC2. So they showed increasing UMOD showed better response, but that was an observational study. This is the thick ascending limb cell. You've got NKCC2, ROMK, and a few transporters. UMOD is a GPI anchored protein. So this is a protein which is anchored on your cell surface with a GPI anchor. You need a protease which cleaves UMOD, and that's released into the lumen. It polymerizes and flows into the lumen. The specific blocker of NKCC2, as I mentioned, was furosemide. So we've got funding for a clinical trial, because we got even before fully understanding the mechanism of UMOD, we can actually have a clinical application. So you got high blood pressure, which is a big problem treatment. Loop diuretics, that's furosemide, comes under this classification of loop diuretics. They are not antihypertensive drugs. The reason being, loop diuretics make you lose sodium so quickly, your blood pressure drops, then your renin-angiotensin system gets activated, and your blood pressure rises up. So it's not a good hypertensive agent. We said that, well, if there are people who are uncontrolled hypertensives, if they have the UMOD increasing genotype, then perhaps in their treatment algorithm, we should bring a loop diuretic early on. So that's a clinical question we want to ask with genotype. So we are doing a genotype-directed clinical trial. It's a multi-center trial. We are going to recruit patients by genotype, give them toracamide, which is a long-acting version of furosemide, and then they'll take this drug for 12 weeks, and we'll measure ambulatory blood pressure, collect some samples, and the trial has just started, and we'll see what happens. But if it shows a significant response to blood pressure by genotype, then we can change guidelines right now, even without understanding the full mechanism by which UMOD interacts with NKCC2. And we got other spin-off things with UMOD. Now we got the UMOD knockout mice and wild type mice. If we feed them salt, now UMOD knockout mice, they do not absorb salt through the tubules, and which means we could study. So what we're doing is we're studying metabolomics and we're studying RNA-seq under normal salt and high salt in both the knockout and the wild type mice, wherein we can use the, if you take the treatment which is salt, you take the genotype which is the UMOD knockout or UMOD wild type, then if you do a genotype treatment interaction, you get a specific effect of salt, meaning if you give salt, you always have noise, you always have the background variability in your gene expression, and we've done this and we found, so if you take genotype treatment interaction, we actually found the acyl carnitine pathway to be the most enriched, which is specific to salt. If you look at the genotype alone, you get a whole host of pathways. You look at salt alone, you get a whole host of pathways. You do an interaction. So we're trying to dissect this further. If you do RNA-seq, we found that salt stress results in heat shock protein being activated. Again, it's interesting because that could be a mechanism by which salt increases UMOD excretion, because we don't know why UMOD production should increase if you consume salt. So yeah, these are some of the spin-off studies we could do. We could actually understand a lot of salt physiology by using this study design. So the next experiment I did was moving on from this to do metabolomics, and this happened through a collaboration with Twins UK. So Twins UK is a large twins cohort based in London. They've got about 10,000 twins, and they've got all samples. And because they are twins, they have done everything: genomics, transcriptomics, anything you want, they have it. So they approached me and said, do you want to do some blood pressure work with them? So I said, fine, and we did metabolomics. So this was metabolome using 200, and they've had the older panel, which is 280 metabolites. We studied about 3,900 individuals with metabolites. So 280 metabolites, we got about 69 metabolites, which was experiment-wide significant, adjusted for everything. So this was a problem. I mean, you had the usual culprits. You had cortisol, you had caffeine sitting there somewhere. So it was a load of metabolites, 69 metabolites. There's nothing much we could do. We then reduced the 69 to 15 independent metabolites. These were quite significant, and because all these metabolites are correlated with each other, we could reduce it to a few. And I've just highlighted a few, three important ones. Caffeine increases your blood pressure. Dihomolinoleate was the most significant of all the metabolites, 10 to the power of minus 17, both for systolic and diastolic blood pressure. And then you had cortisol, and you had a few other fatty acids. And this had one more fatty acid here, which was, I'll come to that later. So this was fine, but what you want is, for me, this was okay, but it was not going to lead to anything, because you need evidence that this is actually causal. The assumption was, if you're an overproducer of the metabolite, then and if that increases your blood pressure, then you have lifelong exposure to high blood pressure. And as I mentioned, even a 2 millimeter change in your blood pressure can affect risk. So luckily the twins had mortality data. So we said, okay, fine, if these metabolites are really going to increase your blood pressure, causally related to blood pressure, then it should kill you earlier. So we related this to mortality. And what would reassure all of you is caffeine does not kill you. It does increase your blood pressure, but it doesn't kill you. So this is just the environmental effect. If people have drunk the caffeine, blood pressure goes up, and we detect that. Dihomolinoleate, as I told you, was the most significant one. Did not have a concordant effect on mortality, actually reduced mortality. Whereas the only one which showed a concordant relationship between blood pressure and mortality was hexadecandioate, which increased blood pressure and increased mortality significantly. So we thought we'll focus on hexa. This is a dicarboxylic acid. And we discussed with various people, and they finally said you still need to prove causality. So the best thing was to feed some rats with hexa. So we got a bottle of hexa from Sigma and then fed these rats, and we measured blood pressure, and we measured hexa levels rose and blood pressure increased. Then we extracted the mesenteric arteries from these rats, and we looked at vascular reactivity to noradrenaline, and we showed that the rats treated with hexadecandioic acid had a greater responsiveness to noradrenaline. So this meant that this was a vascular pathway. So you had a dicarboxylic acid working through a vascular pathway affecting blood pressure. Whilst we were doing these studies, a Canadian group published a paper. They were looking at pulmonary artery hypertension. So they took lung tissue from pulmonary artery hypertension and did metabolomics on them. And one of their top signals was hexadecandioic acid. So that reassured us, because we were thinking it's a vascular pathway that we discovered. We were studying systemic hypertension. So we were studying a systemic vascular bed, systemic hypertension. These people studied a separate vascular bed, the pulmonary vascular blood, and they found hexa. So that sort of helped us a bit, because we thought that, so we used a completely different method to try and establish the validity of a signal. And now we got this, so that helped us, and now we think it's a vascular pathway. So what is dicarboxylic acid? How does this happen? Or how is this produced? Now, most of your fatty acids are metabolized through the beta oxidation pathway, which is in the mitochondria. Only when the beta oxidation pathway is overwhelmed, then you get the omega oxidation pathway, which occurs in the peroxisomes. And omega oxidation pathway consists of 3 key enzymes. One is the CYP4A. Then you have alcohol dehydrogenase and aldehyde dehydrogenase. And this was very interesting, because alcohol we know increases blood pressure. We know that the East Asians, Japanese and Chinese, majority of them have the variant that makes them intolerant to alcohol. Jivas of blood pressure in Europeans did not show any signal with the alcohol dehydrogenase gene, but Jivas of blood pressure in Japanese and Chinese, one of the top signals, alcohol dehydrogenase. And alcohol dehydrogenase is sitting in your omega oxidation pathway, so that helped us. And because I do genomics, we did a GWAS of hexadecandioic, and we found the cytochrome 4 as one of the genome-wide significant signals. But we were very surprised when we found the top signal was SLC01B1. So that's 10 power minus 70. I mean, that means it's not confounded or anything. But SLC01B1 is an organic anion transporter. We found the same SNP, which is associated with statin-induced myopathy. So that's the same SNP which we found associated with hexa. So I think this is not causal. So the problem with statin-induced myopathy is you have a variant in this gene which prevents statins from being absorbed by the liver. So you have more statins circulating in the circulation, it goes into your muscle and causes myopathy. So this may just be the same thing. You got the variant which is not internalizing or absorbing hexa from the circulation, so you're getting higher hexa levels. That's my feeling. We haven't proved it. We need to prove it. We are now in the process of doing a few studies. We've done some gene expression work because the twins, they've got adipocytes and LCL and fibroblasts. So we've done gene expression between these cells and circulating hexa, and we found the top gene, well, rather we found the best signals only for adipocytes, not for LCL or fibroblast skin. We showed alcohol dehydrogenase to be the top gene expressed in the adipocytes, correlating with hexa, circulating hexa levels. So again, that fitted it well. What we are thinking of doing next is, as we think it's a vascular pathway, your vasculature has got perivascular fat. So one way of studying this could be to strip perivascular, so do these vascular experiments in native blood vessels, and we strip perivascular fat and do that, and we can interfere with most of these enzymes with drugs. For example, alcohol dehydrogenase, antabuse, disulfiram is a blocker of alcohol dehydrogenase. There's a blocker for aldehyde dehydrogenase, and for CYP4A, there are blockers. Fibrates, bezafibrate are blockers for those. So we've got agonists and antagonists for that, so we can do a set of experiments, and that's what we will be doing in the near future. As part of all this, we've been doing some metabolomics of drug response through a different grant. And this was, we collaborated with a few US trials, so based in Florida and the Mayo Clinic, and they had stored samples. So this is metabolomics of drug response to hydrochlorothiazide. It took a lot of work, but we got one signal, which was a dicarboxylic acid. So these we just completed. It took a lot of work to get this out, but for hydrochlorothiazide, we are finding that the only signal we have is hydrochlorothiazide. We've done the analysis for beta blockers, and there's no signal at all. So those are two examples of. So it's a lot of work. The discovery part is actually the simplest. Doing a GWAS or doing metabolomics is the simplest part, because you can do the analysis, you get a signal, but translating that to function and trying to make it druggable, find a new drug, that's a long process. That's normal science. You have to go through the whole grind to do that. But precision medicine is also big data. So as you're working on this diabetome project, I'm going to discuss something about the Glasgow blood pressure clinic. As I mentioned, this was a database which started in 1969, when the consultants at the clinic decided it would be a good idea whenever patients come to log all their readings. And at that time they had a computer the size of a room, and now it's all in a NHS database. So they set the process in motion, and they got people to enter the data. So every time a patient comes to a clinic, their blood pressure, what drugs they take, they're recorded. And that's been accruing over many years, and we now have 17,000 patients. We got all clinical data recorded at each visit. We've linked them to the hospital biochemistry and hematology data. We've linked all our patients to the Registrar General to record deaths. We've now linked them to all the hospital admissions. So if any of the patients attending the blood pressure clinic get admitted to any hospital in Scotland, we will have data on their diagnosis, what treatment they were given, and outcomes. We also linked our patients to pharmacy, because as the NHS in Scotland, your prescriptions are free. So if you're prescribed a drug, you go to pharmacy. If a doctor prescribes a drug, you go to pharmacy and you get your drugs free. Now that pharmacy refill is linked to our database. So every time patient picks the drug, we get the data. We've also done the usual thing. We've geocoded. So because our clinic, patients come to our clinic, we have linked it to the meteorological towers, and we got data on various pollutants and weather data. So we did a few studies. One is about weather and blood pressure. Now we know that when people from Scotland, they travel go on holidays to the Caribbean, and they're hypertensive, they start collapsing in the Caribbean, because in the warm weather your blood pressure comes down. In the cold weather you got vasoconstriction, and it's also a thermogenic mechanism, so you got vasoconstriction and your blood pressure goes up in the cold weather. We actually did a complex study wherein we looked at the time the patients came to our clinic. If they came twice within a year, we looked at their blood pressure difference between the warmest and the coldest time of the year, and we showed almost a 6 millimeter or a 6% rise in blood pressure if the patient came in the coldest month of the year. So if they came 2 visits on the warmest period of summer, first visit in the summer and the second visit in winter, there was a 6% rise in blood pressure, which was over and above the treatment-induced drop in blood pressure that we induced at the clinic. But then when we looked at the data, we found that there were 2 types of patients. We had patients whose blood pressure was sensitive to the weather, and patients whose was not. So meaning there were patients whose blood pressure increased in the cold, and there were patients whose blood pressure did not change in the cold. And we showed that if you're temperature sensitive, you have a higher mortality than if you're temperature non-sensitive. And that fitted in with your autonomic system blood pressure variability being a big marker of risk. So that was one study. The other study, because these are all existing data, we're just analyzing existing data. When a patient comes to clinic, we measure blood pressure. They come on a different day, we measure blood pressure. But the blood pressure is never constant. Every visit you come, there's slight variation in your blood pressure. You can calculate the visit-to-visit variability in blood pressure. And it's a long story, but there's a lot of data which said that this variability in blood pressure is a big predictor of stroke. Not your true blood pressure, but the variability. Now, there's data coming out from Oxford, and lots of people have studied in the past, but we felt that was all biased, because if blood pressure is variable, if patients are attending a clinic and blood pressure is variable, that means the people are not taking their drugs. So if they're not taking their drugs, they'll have variable blood pressure and they'll have higher blood pressure, so they'll have a higher risk of stroke. So I wanted to disprove that. So I looked at this study. I did this study wherein I looked at calculated variability in the first year after presentation to clinic, then year 2 to 5 and 5 to 10. And the reasoning is, in the first year when the patient comes to clinic, we are actually inducing variability, because the physician is trying to reduce blood pressure. We are trying new drugs, the patients get side effects. So it's sort of an artificially induced variability. Whereas year 2 to 5 and 5 to 10, patients are attending a clinic regularly, we have established them on drugs, they're happy with the drugs, so it's unlikely that they would be less adherent to treatment. So the hypothesis was, if blood pressure variability is rather the risk that we are seeing with variability is because of patients not taking their drugs, then we should see an association with year 1, but not with year 2 to 5 or 5 to 10. But instead we found it was true. There was blood pressure variability was an independent predictor of death, both all-cause death and cardiovascular death. The higher your blood pressure was variable, you had a greater risk of death, and this was independent of your blood pressure. So I could treat a patient and get the blood pressure down, but if that patient's blood pressure was normal but variable, they had a higher risk. So that's the blue boxes. This is increasing variability, and if your blood pressure is controlled to less than 140, you still have higher mortality. So, but we don't have drugs to reduce variability. We don't know what to do with this. We have no idea. As I mentioned, this correlates with the weather story, because if your blood pressure changes with the weather, that's variability, and that increases mortality. Here we're just looking at variability over long periods of time, and that increases mortality. So nobody knows what to do. Hopefully we have to find a drug which can reduce variability. This is, again, in short, we looked at circulating sodium chloride. Sodium and chloride are very correlated. Salt is sodium chloride, and in your circulation on your body, chloride is the normal anion for sodium. But when we looked at sodium and chloride separately, we found that sodium had no effect on mortality, whereas chloride, lower chloride, was significantly associated with mortality. And this is chloride within the normal range. So if you have low chloride, you die earlier. It took a lot of effort for us to get this published. Nobody believed it. It was rejected, and then we fought with the editors, and finally they published and had an editorial commentary which actually said this is true. And this has been supported subsequently by a lot of data. After we published, this is almost like the rediscovery of chloride. We published this, and lots of people from Cleveland Clinic, they showed a lot of relationship between chloride and heart failure mortality. A big critical care unit in Harvard, they studied 1 million patients, and they showed that low chloride was the best marker of outcomes from a critical care unit. So chloride seems to be doing something which you don't know, and it's not really correlated with sodium. And we got a fellowship. Based on this, we got a fellowship, and we're doing some work on that. The best genetic test for coronary artery disease is a family history. When we look at family history, we assume that if you have a strong family history of heart attacks, you'll be referred to a tertiary care clinic earlier. And in this paper, we showed that yes, people are referred earlier, they are referred at a younger age, at a lower risk of side effects, but they still have higher mortality, which means even if you're recognized, if your GP recognizes you to have a strong family history, you're still at a higher risk. The damage is actually happening much earlier in life. The last one, penultimate one, is diabetes. I told you before that if you have high blood pressure, you are at a higher risk of getting diabetes. One of the reasoning is if you have high blood pressure, 2 of the drugs we give you, beta blockers and thiazide diuretics, actually increase your risk of diabetes, because they affect your insulin secretion, or they affects potassium, which in turn affects insulin secretion. Anyway, those two drugs increase your risk of diabetes. The question is, are they just unmasking diabetes? Meaning giving you a beta blocker or a thiazide is actually a stress test for diabetes. It unmasks and actually recognizes diabetes earlier and you get treated earlier. So the big debate about this, whether this was we were actually inducing diabetes and killing patients earlier by our drugs, or was it just an unmasking of the disease? So we showed in our data that actually this was again got an editorial because it showed something new. What we showed is if you develop diabetes within the first 10 years of diagnosis of hypertension, meaning you came without diabetes but developed diabetes within first 10 years, you are at a much higher risk. If you develop diabetes later on in life, you actually don't have any excess risk. So which means that if somebody is diagnosed with hypertension, we need to differentiate. Are you a patient who's going to get diabetes within 10 years? Or are you somebody who's not going to get diabetes within, are you going to get late onset diabetes, which is of no risk? If that's the case, then I think these people are people who have pre-diabetes. We need to find the right way to diagnose them. And these people are, the late onset diabetes are basically, you've got high blood pressure for a long time, there's vascular damage, so it affects your pancreas and your insulin secreting cells, so you'll get a bit of diabetes later on in life, but it does not kill you. Whereas early onset diabetes is something that we want, so we're designing a clinical trial. The only way to answer that is a clinical trial, so we'll do a clinical trial wherein we'll select pre-diabetics and actively reduce their blood sugar. And we have a placebo group and then try and see if that would reduce. So if we can delay diabetes, will that improve? The question is, if we delay diabetes, then will that improve your outcomes? So that one we are in the process of developing. We also showed, this is a different, I won't go into this. We also showed that couple of antihypertensive drugs that we prescribe, calcium channel blockers and beta blockers, increase your risk of depression. The relationship between calcium channel blockers and depression was unknown. The reason we studied this was Divas of bipolar disease. The top signal for bipolar disease from a Divas was a calcium channel, the target for calcium channel blockers that we commonly prescribe. So it's very complex, but the interesting thing here is we can actually do a natural experiment, because the other drug for hypertension is ACE inhibitors. Now, in pregnancy, if you develop hypertension in pregnancy, the drug of choice would be beta blockers or calcium channel blockers, because the other drugs damage the fetus. So they're contraindicated. So you cannot give them. You can only give calcium channel blockers and diuretics. So to address and perinatal depression is a big problem and is very understudied. So to answer that question, what we are doing is we thought we could do a trial wherein we measure mood, we develop an app. They can, because pregnant women are very difficult to recruit for trials. So we get an app and they answer the depression questionnaire sequentially. Hypertension, we have no choice. We have to treat them with calcium channel blockers or beta blockers. But once they deliver, the ACE inhibitors are not excreted in the breast milk, which means we can now randomize them to remove the CCB beta blockers and give them an ACE inhibitor and then measure their depression. Because if that's the case, that our hypertensive drugs is actually aggravating peripartum depression, then there could be a case for treating this. So what we need to do is we need to achieve the greatest benefit for the largest number at the lowest cost. We need to discover new drugs. We need to make patients take the drugs. We need to diagnose people early. We need to try and screen people and make them realize that they have a problem. The treatment is not drugs straight away. The treatment could be lifestyle measures, reducing weight, reducing diet. All that requires recognition, and that's going to be a big problem. If 1 billion people, we can't do that manually. There are not that many doctors to do it. Even if you get all the nurses, you can't. So we need to do something, and that's why we are thinking of developing you need an app. You need a personalized healthcare app which will bring in your genomics, your lifestyle, your treatment, your risk factors, everything together, and basically make you treat your blood pressure yourself. Because it's very straightforward. It's all protocol. You don't need a doctor once you're diagnosed. You need to take care of your health. And when you take care of if you take care of blood pressure, you're taking care of your global cardiovascular risks, not just blood pressure. Finally, this is just to mention Biobank. Biobank is a 500,000 cohort which we have in the UK. The data is available for any bona fide researchers. There's a lot of extensive phenotypes available. There's genotype. We'll have GWAS data on all the 500. We've got 150,000 GWAS and 150,000 available now. The remaining would be available by spring. You can go to the Biobank website and get all the data that you want. You can look at all the phenotypes, the distribution of the phenotype. You can select people that you want. And it's just a simple procedure. You just apply and prove that you're a researcher or a commercial company. They will charge you for the data. But they are very open to, not collaboration, they're very open to people using the data for developing new insights. But the useful thing is they have got additional phenotypes. They have, in addition to the standard phenotypes, they've got spirometry. They have done in certain subsets, they've done hearing tests, vascular reactivity, eye tests. They've got retinal images. They're also doing MRI on a subset. So huge amount of phenotyping done. But this is a sort of a baseline cohort. This is a biobank, so they were not selected for any disease. This was powered for follow-up and incident events. So they know that they are enough power to get cardiovascular disease, diabetes, colorectal cancer over the next 10-year period. They've also done other things like accelerometer data for physical activity. They are doing a lot of biochemistry now, so we'll get all those results in the next year. They are doing bone scans, and I mentioned MRI, all in subsets of. So it's a huge cohort of quite valuable data which is available for researchers now. And what are we doing in Scotland? So this is the hospital I work in, the Queen Elizabeth University Hospital. But what we have there is we are big on precision medicine. So what we have is we've got the NHS, which is basically the entire health records of the whole country. We've got industry partnerships, which I'll come to, and we've got the university. So we've got all sitting together. And we've created this ecosystem for precision medicine, which is based around the Stratified Medicine Scotland Innovation Centre, which is a government-funded innovation centre which sits on the top floor of a hospital. We got funding from Thermo Fisher in kind, so they've got they basically come in kind to the innovation centre so that they're sequencing 6 exemplar studies for precision medicine. We've got a big data informatics company called Aridia sitting there. And what they are doing, they are integrating all the healthcare data into a secure system where researchers can use the data, do the science, at the same time in a controlled fashion, link the data to back to the patient, because that's important for doing. And the aim is essentially to do clinical trials across the whole country. So you got the whole data from the whole country, you can pick the right people and do a trial from one site. Sorry, I'm going to stop there, and I'm happy to take some questions now or at the end.